Hello and welcome to part 2 of chapter 3's lecture on microscopy and cell morphology. So in this half we're going to talk about the parts of the prokaryotic cell. We're going to look at bacteria and work our way from the outside to the inside, looking at all of the different structures and the function of each of those structures. So I'd like to start with just first some general information about bacteria. First of all, bacteria come in 3 primary morphologies. If you're not sure what that means, a morphology means shape. So we have 3 basic shapes of bacteria. We have cocci or coccus, which are perfectly nice little spheres, nice little round guys. Think staphylococcus or streptococcus. Then we have bacillus. A bacillus is more often known as a rod. That's kind of the general term for it, simply because there is a genre of bacteria called bacillus, so it can be confusing. But we call them bacilli, are known as rods. And then the last are the sporillum. And the sporillum are kind of a bit of like a corkscrew type look. They look kind of like a curly tail. There are also 3 what we call primary arrangements. Arrangements are how the individual or planktonic bacterial cells interact with each other. Sometimes they like to pair off, and so they form these 2 pairs that you see here. And so this is referred to as a diplo. Then we have what's known as the staff arrangement. In the staff arrangement right here, they cluster together, think like a group of grapes. And then the last arrangement that we see is a strep arrangement. Think strep throat, which is caused by Streptococcus pyogenes. And the strep arrangement is usually cocci, although we also see this in rods, in bacilli as well. And they line up end to end and create these big long chains of bacterial cells. There are a few exceptions and some additional morphologies to bacteria. We have here what are called coccibacilli. Coccibacilli are rods that are really short, and they're more of an oval shape. You can see they have kind of an oval shape as opposed to being perfectly round. And so this oval shape, they're actually a rod. They're actually a bacillus bacteria, morphologically in their morphology or shape, but they are sometimes mistaken for cocci under the microscope because they are so short. So when you look into the microscope and you see something that's kind of oval shaped and you're looking at it going, I don't know, is that a rod? Is that a cocci? 9 times out of 10, it's what's called a coccibacilli. That particular morphology is kind of in between a true bacilli and a cocci. Then we have a spirochete. A spirochete is just an extreme form of a sporillum. We looked at a spirochete when we were looking at syphilis in the dark field microscopy. Syphilis is a spirochete. Vibrio, as in like Vibrio cholera, are very long rods that are bent and have this kind of half moon shape, and so this is referred to as a vibrio shape. And so the bacteria that causes cholera has this vibrio shape, so it's actually called Vibrio cholerae. And then down here we have what's called a sarcina arrangement, and this is where this is usually within cocci, and these right here are sarcina. This is actually a picture from a different bacteria, but the sarcina were part of it. So I just wanted to show you what they looked like. So these sarcina here are where we have a whole bunch of cocci that line up, and they usually form these little cubes, and they will either be 4 or 8. So there'll be 4 of them together, and then they'll put 4 more on top, and it'll form a cube-like structure of 8 different bacteria cells. Now, as far as cell structure goes, we are going to work our way from the outside of the bacterial cell to the inside. And here we have a TEM, right? So this is transmission electron microscopy, looking at the inside of the cell. And we are going to talk about pili. We're going to talk about the cell wall. Where is it? Down here. We're going to talk about the flagella that some bacteria have, not all. We're also going to talk about a modified pili or pilus known as a Fimbrae. And then we'll start looking inside of the cell at the cell membrane, the cell wall. We'll talk about the cytoplasm, what makes up the cytoplasm, and of course the genetic material. So let's take a look at the outside. So in the outside, these are external structures or appendages that we find on different bacteria. The first are the Fimbrae and the pili. Fimbrae and pili are both used for attachment, but pili has a secondary function. So Fimbrae are these long tubes. So here I have my bacteria cell. And so these guys, they look like cilia, but bacteria do not have cilia. They just have these kind of hair-like structures that are sticky, and they use these structures to stick to surfaces, to attach to things. And this becomes important in things like biofilm formation and pathogenicity. But sometimes some of these Fimbrae will modify themselves, and they actually become a modified form of Fimbrae called a pili or pilus or sex pilus. And the pilus elongates, and it hollows out, and it becomes this long hollow tube that is going to come into contact with another bacteria cell. We'll just put some Fimbrae on this guy. And so what this guy's going to do is he's going to take a piece of genetic information called a plasmid. He's going to take some genetic material, and he's going to pass that genetic material along that hollow tube into the neighboring cell. And now the neighboring cell will get a copy of that plasmid, of that genetic information. So the pili is used in a process known as conjugation. It's a form of gene exchange or genetic exchange between two bacterial cells. It is what we call horizontal gene transfer. It's called horizontal because it does not result in offspring. I started out with 2 cells. I end with 2 cells. I didn't create any new cells. I just simply gave one cell a new characteristic by giving it new genetic information. So we'll go into more detail of conjugation and how that works in Unit 2. The next structure found on many bacteria are flagella. Now, flagella we think of like in sperm. Sperm are flagellated. Those are flagellated cells in the human body. But bacteria, more often than not, have multiple flagella. And the flagella they have the same function and purpose in bacterial cells that we see in like sperm or other flagellated cells like flagellated protozoans. However, the structure of them is a little bit different, and we will go over the structure in a minute. The last are capsules. Capsules are protective sugar coats that are found on the outside of bacterial cells. The bacteria, in order to produce a capsule, have to have a gene for it, so not all bacteria are capable of capsule production. I call capsules kind of Harry Potter invisibility cloaks. And we'll go into more detail on those as well. So here's our flagella structure. And you can see here's flagella. This is what we call a peritrichous type of microorganism. E. coli is covered in flagella. It has lots of them. Bacteria that are what we refer to as motile, meaning they have motility, will have flagella, and flagellated movement is how they work. The flagella has 3 components. It has a filament, a hook, and a basal body. The filament is composed of a protein called flagellin. This protein is bacteria specific. Human cells and eukaryotic cells do not produce flagellin. This will become important when we look at immunology. Our immune system recognizes flagellin as foreign. So our immune system can tell that it's a bacterial cell that's present. So bacteria use a protein called flagellin to create the filament. The filament is attached to a structure called a hook. This hook allows for, here's the hook here, it allows for rotation of the flagella. The flagellin protein and the composition of the flagella causes the flagella to have a very stiff type of structure to it. I use the analogy of a cat's tail versus a dog's tail. So your cat can wave their entire tail and move it all around. It's very flexible, and they twitch their tail at you and that sort of thing. So that's flexibility within the entire filament. Bacteria have a filament that's more like a dog's tail. When a dog waves its tail, the whole tail moves. They can't just twitch just the end of their tail. So the tail is one long structure. So this filament, what the bacterial cells do is they rotate. So this filament is going to rotate, and it'll turn clockwise and counterclockwise, and this creates movement called rolling and tumbling. That rolling and tumbling feature is when we see bacteria kind of spinning around under a microscope. I'll find a video for you on YouTube, and I'll put that in the D2L course shell so you can see what motility looks like in bacteria. Now, attached, the hook is really just kind of a structure for rotation, allows for that rotation of the flagella for rolling and tumbling to occur. But energy is needed for this entire process. Plus, we have to anchor all of these flagella, they have to be anchored to the bacterial cell as well. So that anchoring is carried out by what's known as the basal body. You can see here that the basal body is underneath the outer lipid layer. This here is the cell wall. This structure right here is the cell wall. And the basal body is actually in the true cell membrane beneath that. This is a gram-negative cell. And the cell wall helps to anchor this here. You can see there's some proteins on either side of the bottom of the basal body, and that is because for energy, bacteria use an electron transport chain and produce energy for the flagella using the proton motive force. You may not know what that is right now. We're going to talk about that in chapter, I think in unit 3 when we talk about metabolism, but it should be familiar to you from your anatomy classes. Capsules are external structures that are excreted by bacteria. Now, in biology, we have a general term for an outer coating, and that's called a glycocalyx. So glycocalyx in biology just means outer coating. There are different types of glycocalyx. There are protein glycocalyx, sugar glycocalyx, and what we call slime molds or slime layer glycocalyx. And bacteria, for the most part, use the sugar one. So they are excreted by some, and this glycocalyx out here, this sugar coating referred to as a capsule, aids in attachment. It's a little sticky, so it's sugar, so it allows to the bacteria to attach more easily. Contributes to, it's considered what we call a virulence factor. It contributes to the pathogenicity or the virulence of an infection by this bacteria simply because of this sugar coating. So here we have some bacteria that are stained. The background is this purple pink you see here. The bacterial cell itself is purple. I've just covered it up with red, but the purple. And then this clearing here, this white area that's surrounding all of these different cells, that's the actual capsule itself. This staining method stains the bacteria in the background but does not stain the actual capsule. Here's another one right here. This one's much smaller, much harder to see. It's just a different staining method. But the idea is that we can see these actual capsules here, and you can see they're excreted on the outside of the cell. They aid in virulence because all of the signals that our immune system would use to recognize this bacteria as something foreign is now covered up by this glycocalyx, by this capsule. So these bacteria are still metabolically active. They can produce toxins, they can take in nutrients, they can carry out all of their normal activities. They just do so in this candy-like coating, this smooth outer sugar coating that our immune system can't get past in order to recognize the cell. An example of a capsule-forming bacteria in virulence is Streptococcus. Streptococcus pyogenes is a capsule-forming bacteria that causes strep throat. So think about how bad, how much it hurts when you have strep throat. All of that inflammation and pus formation, that is your immune system responding in a really potent, strong way. That's what all of that inflammation is. And it's doing that because it knows there's a pathogen there, but it can't get to it. So the pathogen continually signals that inflammatory signal, and the immune system is responding to it, but it's not getting rid of the bacteria. This capsule also allows for immune system evasion. The immune system antibodies can't attach to these guys. Phagocytosis, we have what's known as receptor-mediated endocytosis from our phagocytic cells. They can't carry out those processes properly. The signals that would normally attach to these bacteria to signal phagocytosis and destruction are not going to be able to gain access. So it's considered a true virulence factor. It's also very organized. So a capsule is an organized glycocalyx, and if the glycocalyx is kind of slimy and very unorganized and more liquid-like, then it's called a slime layer. Some water bacteria or aquatic bacteria will use slime layers, not capsules, and they'll use that for attachment and as a form of motility to kind of slide along grass blades and other surfaces. So you can see down here, here's a bacterial cell. The cell wall is in here, but this purple area is the slime layer on, or a form of capsule on the bacteria itself. I call capsules Harry Potter invisibility cloaks. If you like Harry Potter would put on that invisibility cloak and he could run around the castle and cause all kinds of trouble, but nobody could see him. And that's what the capsule is like on bacteria. These bacteria put on their little capsule invisibility cloak and they can run around the body and wreak all kinds of havoc, and our immune system has trouble finding them because they're hiding in that glycocalyx. Now, cell wall. The cell walls of bacteria come in three flavors, you could say. The cell wall itself is a very rigid structure. It's used primarily to maintain the shape of a bacterial cell, and it does provide some protection against pressure and other bacteria. But it's active in some forms of antibiotic resistance. So some antibiotics function, their mechanism of action is to attack the cell wall of bacteria. And if they attack the cell, if the drug attacks the cell wall, breaks down the cell wall of the bacteria, then the bacteria may not be able to survive. But that really depends on the type of bacteria it is and what type of cell wall it has. So we have three basic types of cell walls. We have gram-positive, gram-negative, and then we have a third type that's a very unique class of bacteria. This is actually its own genre, its own genus. And these are referred to as the Mycobacterium. The most famous of the Mycobacterium are the tuberculosis. So TB is caused by Mycobacterium tuberculi. And mycobacteria are very similar to gram-negative bacteria, but they have an additional compound in their cell wall. So I'm going to go through these three little diagrams real quick to introduce you to mycobacteria, and then we'll get more into detail on gram-positive and negative. So you can see here in this diagram, here we have a gram-negative bacteria is first, right? So here's our gram-negative right here. Gram-negative bacteria have an outer lipid layer. That's what you see here. And all right, they refer to as an outer membrane. They have a space, a small space referred to as a periplasmic space. Then they have their cell wall. And the cell wall is composed of a compound called peptidoglycan. And peptidoglycan is made up of two proteins called N-acetylglucosamine and N-acetylmurine, or we refer to them as NAG and NAM. And they alternate, and that's why in this diagram you see green and then kind of this sandy color. So they're showing you the NAG and NAM. These guys are held together, right, by some what are referred to as linker proteins. But if you notice, the cell wall here in the gram-negative bacteria is very, very thin. Underneath that cell wall is the actual true cell membrane. Now if I move over here to the gram-positive cells, two major differences. Number one, there's no outer lipid layer. Okay, so there's no outer layer. Also, look at the size of this cell wall. Since there's no outer layer, the gram-positive organisms have a very thick cell wall. They have layer upon layer upon layer of peptidoglycan, whereas our gram-negatives did not. It's very thin. Then we have, of course, our space underneath, and we have the cell membrane. So gram-negatives have kind of a sandwich-like structure. They have a very thin cell wall in between two lipid layers, and the gram-positive organisms have a very super thick, really strong cell wall with only a single cell membrane or lipid layer. Now over here in the mycobacteria, right, here we have the mycobacteria, they also have the thin cell membrane, and they are like gram-negatives. They have a very thin cell wall. But attached to their cell wall, what you can see here are these glycolipids. And these glycolipids contain two compounds, mycolic acid and arabinogalactan. And together, these create what we refer to as mycolic acid. Now, this mycolic acid makes Mycobacterium very difficult to stain. We have to use a very unique staining method called acid-fast staining in order to get them to take up color. Mycolic acid is kind of like a waxy coating on the outside of the bacterial cells. So that waxy coating prevents Mycobacterium from staining, and we have to stain it very differently. Now back over here to our gram-negatives and gram-positives, this is what we spend most of our time talking about. There was something I forgot to mention that's really important, and that is in gram-positive bacteria, you have these two sets of proteins. One is called a teichoic acid and one is called a lipoteichoic acid. Now the lipoteichoic acids are these protein structures, these molecules that attach the cell wall to the cell membrane, so that everything is kind of attached. teichoic acids just simply stick to the cell wall itself. And teichoic acids are present in gram-positive organisms, but not in gram-negative organisms. And so teichoic acids are oftentimes the target of some antibiotics, and that would make those antibiotics what we call narrow spectrum. They only work on gram-positive organisms because they function by destroying lipoteichoic and teichoic acids. So let's go into more detail, but some better diagrams of our gram-negative and gram-positive. So here's another diagram of a gram-positive cell. This is what gram-positive cells look like under the microscope here. Gram-positive cells stain a dark purple, and they have this very thick peptidoglycan layer. You can see here they've actually labeled the N-acetylglucosamine and the N-acetylmuramic acid. So we have the NAG and the NAM. Those are those purple and blue balls here. Then we have the teichoic acids. Here's your lipoteichoic acid that's running, right, spanning the whole membrane into the cell membrane. And then we have the teichoic acids, which are just linking all of the layers of peptidoglycan together. Then we have our cell membrane down here. This is the inside of the cell. So there's no outer lipid layer on the outside of the cell out here in gram-positive organisms. So gram-positive organisms have teichoic acids that and thick cell walls, whereas gram-negative cells do not. Here are just some other pictures that are showing you where all of the peptidoglycan, all of that kind of stuff is, these different layers, right? So we've got lots of layers here. And this, of course, is another TEM, transmission electron micrograph, that's showing where the difference between the cytoplasmic membrane and where the actual peptidoglycan is. You can see it's very thick here in right in here. You can see that kind of grayish area. It's very thick. So thick peptidoglycan layers anchored together with teichoic acids, no outer lipid layer, and it stains a dark purple in gram staining. Now we take a look at gram-negative cells, and these guys look a lot different. So they have this large, thick phospholipid bilayer, cell membrane on the outside. So this is referred to as the outer membrane or outer lipid layer. They also have their peptidoglycan. You can see the NAG and NAM here, and they have linker proteins, but there are no teichoic acids. So those are non-existent. And then, of course, they have their cell membrane on the inside of the cell. This is the inside over here. And so they have their cell membrane on the inside. In addition to this outer lipid layer, embedded within the outer lipid layer of gram-negative cells is something called the lipopolysaccharide, or LPS. The LPS is made up of two components. They're showing you over here. They have a lipid A, which is embedded into the phospholipid bilayer. That's what you see down here, right? So that's where the lipid A is. Attached to the lipid A is a polysaccharide, a chain of sugar molecules. And attached to the sugar molecules is a long string of sugars that come together to create what's called the O antigen. Okay. The O antigen is going to be very different from one bacterial cell to another. And the best example of O antigens are things like E. coli. So E. coli is named after its O antigen. So E. coli O157H7, the O is coming from its O antigen. What sequence of sugars is making up its O antigen? And so this is the sequence that was labeled 157. The H7 in E. coli comes from the H antigen that is found on the flagella. And so E. coli are highly flagellated, and the H antigen is a protein. It's an H protein that's attached to the outer portion of the flagella of E. coli, then our immune system recognizes. So if we get exposed to E. coli O157H7, we create antibodies against the H7 antigen and against the O157 antigen. There are several other E. colis. There's an E. coli O101, H1. There's an O157H2. So there's a couple of different combinations, lots of different combinations of E. coli in these different instances. Now over here, we can see they're showing you the same kinds of diagrams. We have the membrane, but notice here we have that cell wall in between these two spaces. They're also showing you something called the periplasm. So the periplasm is the space between the two membranes, and the cell wall of gram-negative organisms is located within the periplasm. So it's sometimes called the periplasmic space. We'll talk about that in another slide as well. You do need to be able to label and identify differences between a gram-positive and a gram-negative cell. So in gram staining, this is a differential technique that is used to of course, differentiate between gram-positive and gram-negative cells. It was developed by a scientist by the name of Hans Christian Gram. It uses two stains. It has a primary stain called crystal violet. It has a mordant called iodine. They call it Gram's iodine, but we'll just put iodine. And it has a decolorizing agent called Gram's alcohol, which is really a mixture of acetone and alcohol. And then it has what we call a counter stain, which is safranin. Safranin is that nice reddish color. Gram staining, like I said before in the last lecture, gram staining is the very first thing that's done for the identification of a bacteria. We have to know if it's gram-positive or negative. And the pathogenic abilities of these two organisms differ. I noted to you, I'm going to go back real quick, this LPS right here. This LPS is considered a toxin, and it's found in gram-negative bacteria, so it's oftentimes in the medical field, it's known as an endotoxin, because it is part of the cell, so it's it's inside. This endotoxin does not our bodies do not like this endotoxin, and it initiates inflammation, it initiates fever, and very strong immune responses. So it is a virulence factor that's built in to gram-negative bacteria that gram-positive organisms do not have. So this is one of the reasons that LPS is one of the primary differences pathogenically between gram-positive and gram-negative bacteria. So the gram stain procedure, we looked at this particular picture in the microscopy lecture, but we'll just look at it again real quick now that we know a little bit about gram staining. So crystal violet is our primary stain. It's the first thing that we do. We prepare our slide, we mix the bacteria with a little water, put it on a slide, smear it around, let it air dry, and then heat fix. After it's nice and dry, we then we flood the slide with crystal violet, let it sit for a minute or two. We then remove the crystal violet, not necessarily with water, we just kind of dip the slide and run the crystal violet off, and then flood the slide with iodine. Now when iodine and crystal violet come together, crystal violet is a crystal, it's a salt. So when iodine and crystal violet come together, the crystal violet will begin to crystallize. And if this is occurring inside of that really thick, complex cell wall that we see in gram-positive organisms, then it's going to kind of get stuck in there. With our gram-negative organisms, it just gets stuck in the periplasm. So we have all these crystals forming inside of the periplasm. Once the iodine has sat for a minute or two to allow this, what we call the CVI complex to form, which is crystal violet iodine, once that crystal begins to form, we now decolorize. And in the decolorization step, this is acetone and alcohol. Now for the gram-positive cells, acetone and alcohol tightens up peptidoglycan and causes the cell wall, that thick, intense cell wall, to hang on to the crystal violet, the CVI complex that has formed. But for the gram-negative cells, what happens is the outer lipid layer gets washed away. Alcohol and acetone both dissolve lipids. So that phospholipid membrane that was on the outside of the cell melts away, gets destroyed, broken down. The cell wall that is present in gram-negative organisms is not strong enough to hang on to any of that crystal violet iodine complex. So all of the crystal violet washes out of the gram-negative cells but is retained by the gram-positive cells. In order to see our gram-negative cells, we now counterstain with a lighter dye called safranin. And safranin causes our clear cells to become this pinkish red, and it just intensifies the the dark purple color of the gram-positive cells. So that when we're done, we should see something like this slide over here, where we have gram-negative cells here, and we have some these purple dots here are gram-positive. That brings us to the periplasmic space. The periplasmic space, again, is that space in between the cell membranes. The cell wall is located within it. So in gram-negative cells, we call this the periplasmic space. In gram-positive cells, they just kind of call it an inner wall zone. And it's the space between the cell wall and the cell membrane in gram-positives. In gram-negatives, it's the space between the outer membrane and the cell membrane. This periplasmic space, however, is very, very important. It is important for housing the protons used to create proton motive force and the proton gradient for electron transport chains. Electron transport chains for energy production require a membrane, and the only membrane in a bacterial cell is its cell membrane. So the electron transport chains will all be housed inside of the cell membrane there. And so they're going to need somewhere to house protons in order for the electron transport chain to properly function. It's also responsible for maintaining ionic and water gradients. That electron transport chain, these two are related here. ATP synthesis is because of an electron transport chain. We find periplasmic space in gram-negative organisms and an inner wall zone in gram-positives. That brings us to the membrane itself. The membrane found, the cell membrane of bacterial cells is very similar to animal cells in that it is also a phospholipid bilayer. So it has a hydrophobic and hydrophilic region, or a polar and nonpolar region. Within the cell membrane, we have, of course, different types of proteins. We have integral proteins, like you see here. We have peripheral proteins, which you might see right here. These are integral proteins as well. And, of course, we have channels, which are proteins that allow for the transport of materials across the membrane. The membrane, these proteins are transport. So we have both passive and active transport across cell membranes, and a lot of these proteins are also used for cell signaling and communication with other cells and the immune system. The cell membrane is found between the cell wall and cytoplasm of cells and maintains osmotic pressure. It is semi-permeable. It's a semi-permeable membrane, so osmosis and diffusion does occur across the cell membrane. There are different types of transport. I'm not going to spend a lot of time on diffusion and osmosis. You should have covered this in your Anatomy 1 courses as well as your chemistry course. But just a quick refresher, we have the terms of tonicity. So solutions can be hypertonic, which means the environment has more solute than inside of the cell. Then we have hypotonic, which means the environment has a lower solute, so the cell takes water in. And then we have isotonic, where the environment and cell cytoplasm have the same solute concentration, and so we have equilibrium. So this right here is an isotonic cell. The solution inside is the same as outside, so the cell maintains the integrity of its shape. Then we have a hypotonic solution. In a hypotonic solution, referred to as hypoosmotic, water moves into the cell because the concentration of solutes inside of the cell is higher than that outside of the cell. Because the cell is selectively permeable, those solutes cannot move out and water moves in. The cell will eventually fill up with so much water that it can no longer maintain its shape, and the cell will lyse open. In a hypertonic solution, we have the exact opposite. Our solution here is like putting a blood cell in a 10% salt solution. There's more water inside the cell than there is outside of the cell, so so it has to water will move out of the cell because solutes can't move in. Remember, semi-permeability. In this case, the cell will go through what's known as plasmolysis. Now, in red blood cells, you would call this crenation. But because we have a cell wall The cell will maintain its shape, just the cell membrane will lyse, will shrink up. And this is a problem when we're doing things like microbial counts with a microscope, because we could actually count a cell that we think is an existing cell, but it's really a dead cell. And that's because we don't know that plasmolysis has occurred inside of the cell wall. Now, transport across the membrane occurs in two basic forms, passive and active. And now, in passive transport, and I always like to think of transport in terms of gradients. So it's all about concentration gradients. And you can think of a concentration gradient as a hill. Now, in passive transport, we're going to move with the concentration gradient, which means we're going to move downhill. If I move downhill, then I don't have to put any energy to it. If I have a ball up here at the top of the hill, the ball will naturally roll down the hill without any input of energy. So this is moving with the concentration gradient. Osmosis, movement of water based on concentration, and diffusion, movement of water based on concentration, are both forms of passive transport. There's a third form of passive transport called facilitated diffusion. In facilitated diffusion, we would use a protein to facilitate the movement of something across the cell membrane, but in order for that solution to move or that solute to move, no energy input is required. So there's no, it's protein-assisted movement, but not necessarily with the use of energy. All three forms of passive transport rely on concentration gradients. So the concentration has to be higher in one area than in the other. Now, in active transport, we have just the opposite. We still have a concentration gradient, but instead of moving down the hill, now my ball is down at the bottom of the hill, so I have to push it up. In active transport, I'm going to move against the concentration gradient. And when you move against a concentration gradient, that's always going to require some form of energy input. Now, we say ATP because that's the current energy currency of the cell. Other forms of energy could be used as well. So think about this ball. In order to get this ball back up to the top of our concentration gradient, we would either have to carry it and walk up the hill or push it up the hill. Either way, both of those are going to require some form of energy. Another form of active transport, a type of active transport, is called group translocation. In group translocation, I would have a whole bunch of people. Think about the old-fashioned firehouses, right? So here I have a couple of people, and each person is standing on a different part of my concentration gradient. And what I'm going to do now is, instead of me rolling the ball up the hill on my own, I'm going to hand this ball off to this guy, who's going to hand it off to this guy, who hands it off to this guy, who hands it off to this guy. Each of these people are a protein in this chain. It takes energy to move from one person to the next. And I'm going to move my solute and move it as I go against my concentration gradient. And I'm putting in energy. But when I do this, in order to keep the ball from rolling back down the hill, I may structurally change it. And so when I get up here, the ball is now a square, so that the square cannot freely roll back down that concentration gradient. So that's what's meant by the molecule being chemically altered during transport. As the molecule is moving across the membrane, because a group of proteins are working together to move it, the molecule itself will become chemically altered, so it cannot leave the cell. So here's just some examples. This is passive transport versus active transport. So passive, I'm moving with my concentration gradient. Active, I'm moving against it using energy. Over here is facilitated diffusion. This is also a form of passive transport. And in facilitated diffusion, I have all of these molecules outside. And what's going to happen is my molecule will bind to a transport protein. Okay? Now, this is like a channel. And if you remember from anatomy and the structure of proteins, a protein goes through a conformational change when something is attached to it. That's how proteins work. They work by changing shape. So this protein is specific to this little molecule here, right? This little square. My pen is not working. Let's see what happened to my pen. Let's try changing colors. Sometimes it just kind of freaks out on me. We'll try again. So there we go. So these little molecules here Excuse me, sorry about that. So these little molecules here are specific to this protein, and when they attach to it, the protein changes shape. That change in shape causes the protein to kind of move this molecule across the membrane and into the inside of the cell. And this form of facilitated diffusion, again, is passive. It works by changing the shape of the protein, not by inputting any kind of energy. And then our last form of transport here is group translocation, and I'm going to show you a quick video. Don't worry about all the names of the proteins and such. I just want you to understand the concept of using multiple proteins with the addition of energy to not only move a molecule across a membrane, but to chemically alter that molecule during its movement.
Group translocation is a process in which an organic molecule, such as glucose, is transported into the cell while being chemically modified. The PEP phosphotransferase system is an example of group translocation. In this process, the phosphate from the high-energy compound phosphoenolpyruvate, or PEP, is transferred to HPR by enzyme 1, and the product pyruvate is released. The phosphate is then transferred from HPR to enzyme 2B by enzyme 2A. The membrane-bound enzyme 2C then carries the glucose across the membrane, and the glucose becomes phosphorylated as it enters the cell.
So this is the first step of glycolysis, by the way. And what is happening is those proteins, we're all moving that phosphate group around so that as glucose gets brought across the cell membrane, it gets phosphorylated in the first step of glycolysis. And that phosphorylation is the chemical change that occurs in group translocation of glucose.
Group translocation is a process in which an organic molecule, such as glucose.
So now that we've looked at all of the external structures and we've moved our way across these cell walls and cell membranes of gram-positive and gram-negative cells, we now look at the internal structures. And honestly, there's not a whole lot to look at. Inside of bacterial cells, these are prokaryotic organisms, so there are no organelles. Bacteria do not have membrane-bound nuclei, they don't have mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, none of that. They don't have any organelles. They basically have their genetic material, which is in two forms, chromosome, which is their primary genetic material, and plasmids, which is additional pieces of DNA. They have ribosomes because they have to make proteins, and they have cytoplasm, which is fluid that all of this is floating around in. So the chromosome is their primary, the largest piece of DNA within. Think of our own chromosomes. This is what makes the bacterial cell a bacterial cell. And the chromosome is located, usually it's not anchored, but it's kind of located in one region of a bacterial cell. And that region is referred to as the nucleoid region. And that's mainly because that's just where its chromosome is found. Now, plasmids are interesting. Plasmids are really tiny little circular pieces of DNA. We will talk about plasmids a lot in microbiology. They are unique to the bacteria. And these are pieces of DNA that are in addition to the chromosome. So I have my bacterial cell and it has its chromosome over here, right, all its DNA, but then all of a sudden it has all these little circular pieces of DNA. And these are all plasmids. And plasmids contribute to genetic diversity. So this cell might have these 5 plasmids, but I got a bacterial cell over here, and this one only has 3 plasmids. This guy over here, through a sex pilus and conjugation that we were talking about in the beginning of the lecture, will transfer a copy of one of its plasmids over into this cell, and that's that conjugation that we talked about. And that's how plasmids help to contribute to genetic diversity. So they are transferable between bacterial cells. Then in addition to that, because they move around a lot, they pick up genes here and there from different bacterial cells throughout their lifetime. and they change over time as they get transferred. Copies of them get transferred from one cell to the next. And these plasmids start to pick up different genes. So many of the virulence and antibiotic resistance genes that we see found in bacteria are most often located on plasmids. Ribosomes, we should know from our anatomy classes, ribosomes are those protein RNA structures that are responsible for the translation of RNA into proteins. They're part of the DNA to RNA to protein cycle in biology. And then we have the cytoplasm, that viscous kind of fluid that houses these internal structures. It consists primarily of water, proteins, in both soluble and insoluble molecules. Another internal structure that is unique to bacteria here that we have to mention, this is only produced in two genera of bacteria, but they are called endospores. And endospores cause lots of problems. Some diseases that are caused by endospore forming bacteria, by the way, they are the Clostridia and the bacilli. There is a Corynebacterium that are closely related, excuse me, to the bacilli. They're considered a bacilli. Excuse me. So these gram-positive bacilli, they are referred to as gram-positive endospore-forming bacteria. And endospores are literally spores that are found inside of the bacterial cells. So that's why they're called endo. And inside of that spore, the cell goes dormant. Now these spores, unlike capsules, are protein. They're keratin for the most part. And they start out, we have our normal, what we call the vegetative cycle of a cell. This is a cell that is a bacterial cell that is kind of living its life, right? It's doing its thing every day, making dinner, going to school, doing its everyday thing. But then times get tough, and there's not enough water, there's not enough nutrients. It's a hostile environment. So the cell makes copies of all of its genetic material, a couple of new ribosomes, takes everything and starts walling it off in one region of the cell, starts building this little septum structure out of cell wall and starts surrounding that copy of everything in peptidoglycan, almost as if it's going to create a new cell. But it just keeps building on it, and then it starts coating it, this cortex here, it starts coating all of this stuff with some keratin, this really thick protein coat outside of the thick peptidoglycan, and eventually the parent cell that made the actual spore starts to die away, fall away, and what we're left with is the actual spore itself. Now this spore is a dormant form of the bacteria, and endospores in their spore form, what we call free spore form, can last for years. They're found in soil, in harsh environments, and they can last for an extremely long period of time. They can resist drought and loss of fluid or water for a long period of time. The cell is at its absolute bare minimum metabolism to even survive. But when times get good, right, think of them like a seed. When water is now available and nutrients are now available, these bacteria will begin germination like a seed. And so germination results in new vegetative cells that enter into this vegetative cycle, and then it can begin again. They either go and start creating new colonies, or times get tough and they go back into the sporulation cycle. Why are these important? Clostridia over here causes C. diff infections. So you may have heard of a C. difficile or a C. diff infection. That's a gastrointestinal infection that's really severe and oftentimes causes ulcerations and bleeding. Bacilli can cause, you get it from soil, tetanus, excuse me. Oh, goodness. Long day. Tetanus. Clostridia also causes botulism. That's another one. Bacilli is tetanus. Corynebacterium can cause some GI infections, but it's not nearly as bad, and some skin. So C. difficile, botulism, tetanus, these are some, it's actually Clostridia causes tetanus, excuse me, it's Clostridia tetani. So tetanus. Bacillus is anthrax. respiratory. This is affects the nervous system. So anyway, you can see these are some pretty serious diseases, and they are easily transmissible. Both of these bacteria are found in soil, and Clostridia is found in the GI tract of warm-blooded mammals, including humans. GI tract. It's also found in humans, and C. diff infections are oftentimes what we call superinfections due to overuse of antibiotics. Other internal structures, there's the concept of a newly discovered type of cytoskeleton. It's temporary. It's usually used just for cell division to help with what's known as binary fission. Then we have storage granules. Storage granules are inclusions that are part of the actual cell membrane itself that bacteria will use to store compounds. So starch, iron, phosphorus, nitrogen, glycogen, these are different storage granules. Magnetosomes are used by very few. They're quite rare, but they are used by some bacteria, particularly in aquatic environments, to orient themselves to the poles of the earth so they can move around in different areas in bodies of water. And the last are gas vesicles, also used by bacteria in aquatic environments, similar to the swim bladder in a fish. They use it to control buoyancy. Sometimes they want to be closer to the surface, sometimes they want to be lower down in the water. Now, I'm not going to go over all of the parts and function of eukaryotic cells. You should already know that from your anatomy courses, but you do need to review particularly animal cells. Plant cells we're not going to cover too much in here, Oops. But you want to look at the plant cells, make sure that you know all of the different organelles, the functions of those organelles, and how those organelles interact with each other, because we will be addressing and talking about lots of different parts of the animal cell itself when we talk about bacteria and how they enter into cells, viruses, what viruses do when they get inside of cells, and how they can destroy cells. So it's important to understand the structure and function of a eukaryotic cell in order to understand and derive how bacteria can affect those cells. Now, we already have compared prokaryotic versus eukaryotic cells, but I just want to make sure that you know the major differences. Remember, we have no membrane-bound organelles in prokaryotic cells. They use a different kind of ribosome. It's a little bit different. It's more like the mitochondrial ribosome in eukaryotic cells. There's just some minor differences, but you do want to make sure that you know about and of course, no membrane-bound organelles. So make sure that you are capable of identifying and comparing and contrasting prokaryotic versus eukaryotic cells. So that is the end of our lecture. I do have a couple of review questions that I think you should ask yourself. You want to be able, of course, like I said, be able to label the parts of a prokaryotic cell. You want to know what the functions are of the different external structures in bacteria. You definitely want to know how genetic material in a bacterial cell is different from that in an animal or plant cell. Think structure and location. How are endospores and capsules different? Compare the two of them and what makes them unique. Why are capsules considered virulence factors? What are the differences between gram-positive and gram-negative cells? What do gram-positive cells have that gram-negatives do not? What are the different forms of transport for molecules used by prokaryotic cells? What form of motility do bacteria use? Compare and contrast prokaryotic to eukaryotic cells. And on your own, you need to review that cell structure and function. I don't cover eukaryotic cell structure and function in this class because you took one or possibly two anatomy classes already. And like I said, it's very important to have that. So make sure you understand there's two really important things you want to know. Of course, structure and function of eukaryotic cells. Another one that I barely mentioned, but I do want to drive home is protein structure. So make sure you understand the levels of protein folding and how proteins function, that shape to that conformational change to function. So how proteins function. Just want to add that there. How do they work? What makes a protein work? So these are just some really great review questions for this particular lecture. You can write them down and answer them. We will talk about these in class when we meet. So I hope you have a good evening, and I'll see you all in class.